Welcome. Thank you very much for coming. And I want to say thank you very much for coming not only to the people in the room, but there are lots of people on Zoom who have also decided to attend this. It's a very weird experience, I have to tell you. I really hope this lecture goes well. It's the last shot, right? So we'll see. One of my colleagues at the University of Amsterdam, Henk Hiemstra, gave an exit interview because in there, that's kind of just normal. Everybody does it. And I thought this was an opportunity to look backwards. Universities are great at looking forwards. We got a new hire, we got a new grant. It's about where we're going. And I guess this is kind of the rear view mirror. And what I want to do is, it's a celebration. For me, I don't know about you, but it's a celebration. I hope that's how it'll come out. What I want to try and do is to tell you a little bit about, well, that's not good. Sorry? Which screen? Here we go. Okay, this could not be a normal lecture without technical issues. So just a minute. Now what happens? Oh. No, it doesn't advance at all. It's gone to sleep. Hey. Yay, okay. So you're going to hear a little bit about the good CV. That's the published one. You're going to hear a little bit about the bad CV. That's not the unpublished one. What I'm hoping to do is give you sort of 4 research vignettes that span the 40 years. So literally right now, 40 years ago, I got a letter of invitation to what was the Department of Chemistry, not Chemistry and Chemical Biology, but the Department of Chemistry to join. And so I'm going to tell you a little bit about the research since then. There'll be sort of 4 vignettes split up by 1 comment about teaching and 1 comment about the administration. At the end of the day, partly this is about looking backwards. So 40 years ago, some people sat around in a room. I said, I guess we'll give this kid a try. Did the bet pay off? The other part about this is this lecture shouldn't just be about me telling you what happened. What I'm hoping is there are lots of students in the audience. There's some of my younger colleagues in the audience. I am the voice of experience. I'm not bragging. I have made a zillion mistakes, and I actually learned from some of them, okay? And so if I can share some of those suggestions, they'll always show up in green. You know, accept them or ignore them. That's okay by me, but that's sort of the general idea. Let's give you some historical perspective. I arrived on July 2nd, 1985, to a chemistry department that had 31 white men that were, half of them rather, were educated in England, because that's the only place that good education was done, apparently, in those days. So I looked like that, which is kind of interesting. Fast forward, so that's a picture that seems to be ubiquitous on campus at the moment. So aside from both white hair and gray hair, sorry, and no hair, it's sort of this. I did an experiment first year that here's the rate constant for my hair turning white. Here's the rate constant for falling out. Well, will I be bald or will I be white haired? Anyway, so what happened is that no, we do not yet look like all of Canada, but boy, we've made directions in the right, we've made good choices and we're moving in that direction. So I'm really pleased about those changes. I have to teach you a little bit about silicon chemistry because otherwise it won't make a lot of sense. There will be those who are very comfortable with chemistry, and others unfamiliar. Most people know about organic chemistry, carbon, 4 bonds, life, all that stuff. And we tend to think of carbon as being the normal element. Wrong. Silicon, germanium, tin, and lead all behave like you would expect based on their neighbors on the periodic table. Carbon is weird, okay? Silicon also typically forms 4 bonds, but it shares its electrons very differently. So if you look at a silicon-hydrogen bond, sorry about the wiggling there, the silicon-hydrogen bond, it's not a proton like it would be in carbon, it's a hydride, it's a reducing agent. And the other thing is that, silicon-oxygen bond, this is really bad, sorry. Silicon-oxygen bonds are extremely strong. So when you run into a natural silicon atom on this planet, most of the time it's sand or silicate or something like that with 4 bonds or more to oxygen, because that's where it wants to be. So if you want to make a silicone, for example, what you do is you start off with sand, you get all of those super strong oxygen bonds of silicon, and you reduce them away. And at the very best, you get 1 CO2 for every SiO2 you make. But this is very endothermic. You need 2,000 degrees C to make this work. So in most places on the planet, that's a very heavy source of greenhouse gases. There's a huge carbon footprint for silicone because we have to overcome that bond. Once you've done that, now we go and add some carbon bonds. Sorry, Siri, I didn't mean to say that. Some carbon bonds to silicon, then you add back the oxygen. So it's a dumb process. You start with 4 bonds to oxygen, you end up with 2, but that's commercially the only way that people can do it in a practical sense. And so this is a silicone polymer over here. It looks like an inorganic polymer if you look at it along the chain SiO, but above and below the chain is grease, all these methyl groups. And the weird properties that silicone has compared to organic polymers come from this tension between the two kinds of chemistry. When I arrived, I wrote an NSERC grant, which was funded, and it didn't work at all. And neither did some of our other projects that we started, but you're supposed to have a research program. That's what NSERC is demanding. And so we kind of quickly pivoted to doing something called the beta effect. So the beta effect is when you have a positive charge beta, like 2 carbons over to silicon. I told you that silicon wants to give up electron density. If an undergraduate student had written this primary carbocation here, they probably wouldn't pass the test. But silicon, that carbon silicon bond that's in red, is forming a kind of a partial pi bond. It's dumping electrons into that cation, stabilizing it. People knew quite a bit about this in the organic synthetic community in those days when R was a methyl group or a phenyl group. Well, to try and find something different, we said, what happens if you now use oxygen or chlorine or fluorine? And we were able to do a mixture of kinetic and mechanistic studies that allowed us to show that this was directly related to something called group electronegativity. And like everybody in the room, the world yawned. Okay, great. Thanks for that. But it was a thing, right? And we did vinyl cations and a bunch of others. It stood as a kind of a research program. And one thing that, however, did fit into my training as an organic chemist was this. Here's the beta effect with a silostyrene. You add a proton, you get the beta cation. That's not so great. Then we make a carbon-carbon bond, hallelujah. But even better, the aromatic ring does a Friedel-Crafts alkylation. So one reaction, 2 carbon-carbon bonds formed, and it's stereoselective. Now it starts to feel like what I was trained to do. The world continued to yawn. Nobody could care less. In the academic world, and we're at a conference, and some industrial guys came up to me and said, look, sand likes water. People do a lot of chemistry on sand or fiberglass. You've got to do a modification of the surface of fiberglass if you're going to actually use it in practice. Often they use something called an adhesion promoter or a coupling agent, which binds through a silicon-oxygen bond stuff onto the surface, kind of like this. So that turns what is a water-loving bead of silica into an organic grease-loving bead of silica. That's actually a really important outcome. And industrial people said, mostly these are single point of contact, but your molecule will have 2 feet on the silica, and that's different. And I went, oh, so I could get like industrial money? That had never crossed my mind, yeah? And also that opened this door to application. Some observation you make in the lab might be intrinsically interesting or aesthetically beautiful. It might also be useful, and keep that door open. Here's an example that was done by Ebad. Where's Ebad? Ebad Noman, that guy right there, and an undergraduate student. The undergraduate, Masanji Bio, was mucking around for reasons that don't even matter. That wasn't what we were looking for. I love this. We were taking bromine and mixing it with 2 different kinds of silicone. So up in the box, you see there's sort of silicone in a blue box and silicone in a yellow box, 2 different kinds of rubber with 2 different kinds of cure chemistry. And Masanji adds bromine, and the one in the yellow box disappears in 10 minutes. So here's a way to take 2 kinds of rubber at the end of their life, a complicated mixture, add bromine, and only one degrades in 15 minutes. And the other one takes a day to degrade. It's a kinetic phenomenon, which is great, and it hinges on the beta effect. What's happening is that hydrogen is oxidatively cleaved by the bromine, make the beta cation. I'm one of the few people on the planet that would actually understand this because I had all that beta effect stuff 40 years ago or 35 years ago, and we are doing sustainability. So the reason I want to mention that is because, first of all, get your students to tell you whatever happens, whether they want to, whether it's what you're expecting or not. Often the unexpected ones are the most interesting things. And also let students try stuff. I'll get back to that later on. My mom gave me this poster as a teenager. There's no heavier burden than a great potential. Thanks, Mom. So it was in my bedroom while I was a high school student, and that perfectly describes the wilderness years. I think they are the wilderness years. So I got a starter grant. That was great. And after the first year, they cut me 10% down. Thank you, because they were only 3 years in that time. I got $25K from McMaster. That's so much money. But I had to split it over 2 years. What could you buy for $12.5K? Nothing. I had to throw out the first PhD student after a year because he stopped coming in because he was building a house. So we got 5 papers on our own and 3 papers with some other colleagues inside the chemistry department. And I was funded at the time by NSERC. So I was a University Research Fellow. The money for my salary did not come from McMaster, but McMaster was required to make a tenure decision after 5 years. And they held their nose and said, okay, but you don't get promotion. And that's not allowed now. They basically said, no, you don't cut the mustard quite. Do better. There's no heavier burden than a great potential. So what was the problem? I had no idea how to be a professor. I walked in on July 2nd to the chemistry office, and Kirsten Stockman, who was the secretary for me and 13 of my friends, my colleagues, said, why are you wearing a tie? Nobody wears a tie. That's right, because I didn't know how to be a professor. So I'm taking the bloody tie off now, like I should have taken the bloody tie off before I got there, because I just didn't know. And what else didn't I know? I thought I was supposed to sit in the corner and do good stuff by myself. And I was either too dumb or too proud to go and ask for someone to help me because I didn't understand the system. So I had really no mentors, either provided, and I didn't ask for them. I didn't know what a network was. I thought that the only thing that was interesting was this narrow thing that I set out to do. No, weird stuff is good. So these green things are what I advise all of you. If you're a student, get a LinkedIn profile right now. People will find you, you'll find them. This networking thing is unbelievably important in the academy, in companies, in government. And step out of your comfort zone, which I'm going to talk about a little bit more. All right, so the second part of the research comes back to solving those challenges that I just set. If you don't have mentors, you're in trouble. I didn't have to go find them. They came and found me. So Bob Pelton, who's sitting right there, arrives in 1987, and he's done work in the pulp and paper industry. He's an expert on colloids and interfaces. I'd never heard of a colloid. I didn't know what a colloid was. But he was happy to tell me. He said, there's 2 ways to stabilize one phase and another. If you shake up water and oil, you get a mixture and then it falls back to being 2 layers, right? Well, if you want to make salad dressing, you have to add something that stabilizes the droplet of oil and water. And one way to do it is to make those droplets or particles charged so that as they approach one another, there's electrostatic repulsion. And then the other way is to do what's called steric stabilization. So organic chemists don't use the word this way, but essentially if you put a hairy layer on the surface of a droplet and the hairy layer loves the solvent, that's the big key, then it'll be stabilized. And it's hard for those things to come together and interdigitate, so to speak. So with Bob, what we did was to take advantage of the unusual chemistry of silicones and give the materials a haircut. So if you start off with a particle with these long hairs of silicone, you see if we try to get the particles to jam into one another, there's space. Can't do it. But if you give it a crew cut, what we find is that there's a critical point where the chain lengths aren't long enough to stabilize the particles and they collapse and crash. So here's a case of being able to use this unusual property that silicones happen to come with to answer a sort of, I guess it's an academic question, but it goes beyond that because often we want to stabilize or destabilize systems, and we could get at that through this chemistry. And we also did with Lucy Yee some work using what was essentially electrostatic stabilization where it's charged now. But we did some biological stuff for the people in the audience that think that way. It's biotin and streptavidin. And if we could take a titania particle, I guess I should be looking at this screen, that's better. Titania particle, then we could put biological goo on the surface, give it to a poor E. coli bacterium, and you see we kind of made a rock all surrounding this bacterium. Okay, that was a lovely picture, but if you shine light on that, the titania generates oxygen species that kill the pathogen. So it has function. So again, you know, some kind of fundamental understanding leads to a practical material. Bob also tried really hard to help me overcome my natural proclivity to make really dumb decisions. He's only been partly successful at that, I think it's fair to say. The second person is John Brennan. So John was also interested in silica, but for a different reason. He wanted to be able to put proteins inside silica columns to do chromatography. So most people in the room have actually done chromatography at school. Put a bit of ink on a piece of paper, let the water go by, the ink separates into different colors. But Brennan didn't want to separate these molecules by color or mass or molecular weight or steric size or charge. He wanted to have separation based only on whether the compounds bind to a protein or not. If the compound doesn't bind to the protein, it could never be a drug against the disease associated with the protein. If it does bind to the drug, it could be a candidate. Well, here's the problem. If you start off with tetraethoxysilane, and that's what the world did in those days, and you make silica, you get initially something that looks crystal clear with glass, and it just keeps curing. And then you get shrinkage, and either you get severe cracking across the glass, or you get pull away from the tube that it's sitting in. In either case, you lose your ability to do chromatography, number one, but number two, the proteins were being denatured. So people also know about protein denaturation. If you don't, you actually do. If you want to make a fried egg, you crack the egg, the white is natured. Now you make a fried egg, it turns white, it's rubbery, it's denatured, it's never going back to where it started. Brennan needed to work on proteins that were in their native state to do this drug testing. And so what we discovered also is that if you put proteins in at the beginning, while you're starting the chemistry, you get the silica tube, no shrinkage, happy protein, and we could run chromatography experiments, as I said, to get rid of the losers and hopefully leave then the focus only on potential candidates. The third person is Mark McDermott. Unfortunately, Mark has died in the meantime, and he phoned me up one time, I don't know, in the late 80s and said, all we have to do, be super careful when somebody says all we have to do. Here's all we had to do. Everybody in this room has eaten protein in the last 24 hours, I'm pretty sure. So it's happily digesting in your stomach. Why? The pH is 1.5, and you've got all these enzymes in there designed to convert that food to use inside your body. So all we had to do was get the protein through the stomach with all those enzymes at pH 1.5, and then get it to the small intestine where the pH is 8, and have it be released to find the mucosal immune system. All he wanted was an oral vaccine. That's all. Remember COVID? Yeah. So I was so naive because it turns out the silane business is well known. You can take two functional groups, block one with silicon, turn the green alcohol into a red ketone, get rid of the silicon. We use protecting groups all the time. So what we'll do is we'll take silicone and we'll mix it with a protein. We'll make some kind of particle where the silicone's wrapping around the protein, protecting it in the stomach. The amount of protein you need for a vaccination is too small, so we'll just bulk it up so that we can actually see what we're doing. So we dumped it into starch, and then we'll see whether it's an oral vaccine. Well, the cartoon is pretty good. So Paul Zelisko, who's sitting here somewhere, showed that, in fact, this cartoon is true. You've got a red protein and a yellow protein, and the red one loves the black stuff, which is silicone. We didn't know that at the time. What we then did is make, and Bob's going to laugh at, these are the worst particles ever made on the planet. They're not monodispersed, they're tiny, they're huge, they're ugly. But if you feed them to a mouse, you can see this purple line. The mouse develops an antibody titer against the protein. So it got through the stomach into the small intestine to do antibodies. Everything old is new again, eh? On Gilmore Hall in the first year of COVID, there was this huge banner about all of the inhalable vaccines that were being developed in the hospital. Yeah. These ones, 1993, we all reclaimed and showed inhalable vaccines. So why am I not sitting in my castle in Tahiti, you might ask? Because the answer is we won the Synergy Prize from Connaught Labs, the Conference Board, and NSERC for this work. We got a patent. We have a lot of papers. And they said, because silicone breast implants kill women, because that was a thing in those days. That's not true, but that's what was kind of in the general area. So they just walked away from it. Those are surprises you have to get used to, expect. Don't expect them, but maybe don't be surprised when they arrive. One other thing to thank Mark for, he was very interesting. You know, we wrote this, I wrote the first draft of the application for the Synergy Prize, and it was very scientific and technical. He said, throw it in the garbage. Tell them why it's going to save human lives. So he wrote it, we're going to save the world. We got the prize, okay? So that's another thing. Remember who your audience is, because it can change. The fourth person is Heather Sheardown. So Heather is currently Dean of Engineering. She's a world expert in ophthalmic materials like contact lenses. Everybody who is over 35 stops wearing contact lenses and goes back to spectacles because they're uncomfortable at the end of the day. And why are they uncomfortable? Among other things, because the proteins that are in your tear film go and sit down on the contact lens, which is a silicone hydrogel. So you can see that if you put a drop of water onto silicone rubber, the water kind of beads up. John Brash, also from chemical engineering, a real superstar, showed you can dramatically change the wettability of the silicone by putting this layer of polyethylene oxide on there. But you also can change the facility or affinity of proteins for that surface. So if the proteins don't go down and sit there, two things happen. One, you don't have a rough surface that your eyelid is scratching over, which makes them uncomfortable by the end of the day. And two, you take off the table the chance of denaturation leading to a negative biological event. So what I did with Heather was basically, and Hong Chen, the trouble is this thing is catching my eye. So I need to go like that. Thank you. Okay. This thing essentially is polyethylene oxide, but we put a simple little organic linker on the end. And that linker would react with amines and alcohols. And what happens is that you can now take that material and put onto the silicone surface growth factors, adhesion peptides, proteins, enzymes, heparin. And there's this layer of polyethylene oxide and water that prevents from all that biological stuff from seeing the substrate. The enzymes had essentially the same turnover rate, bound to a surface or unbound. That's very atypical. So again, it's bringing different expertise sets together. Heather was running the 2020 NSERC Research Network on Ophthalmic Materials. This was some of the first experience I had with dealing with doctors and biologists and stuff. And every one of those interactions is great because they know stuff you don't know, but you know stuff they don't know. And that's part of the fun. And the last person I want to talk about is not a mentor. This is one of my students, but it was a lesson learned. So Wei-An Zhao worked for me as a master's student. And then he said, I want to do a PhD but I want to do DNA chemistry. Okay, so I said, go work for Ying-Fu Li. He's like a world expert. And Wei-An said, nah, I want to work for both of you guys. So we cooked up this project and we all sat down in a room, told him, what to go away and do. And 6 months later, we have our first meeting. So Wei-An says, I want to tell you about the gold nanoparticles. And so Ying-Fu and I looked at each other and go, we never once talked about gold nanoparticles. He said, here's the manuscript. And the manuscript was this. You basically take the big orange gold nanoparticles, not so big, and you put some DNA on the surface. So there's the little green squiggles. That's DNA. And then you use the circular DNA and the correct enzyme. And what happens is you grow this long chain from what is the complementary DNA to the circular stuff. And it happens over and over and over. It's called rolling circle amplification. Cool. And if you also encode somewhere in that circular DNA, the complementary DNA for the DNA that's sitting on a gold nanoparticle, which is what he did, you now could put little tiny gold nanoparticles like a necklace. It's a DNA necklace with gold nanoparticles. There's the picture. So every stripe that you see is DNA and the little bumps are gold nanoparticles. It was really cool chemistry. So that's one thing, but it was because Ying-Fu and I said, whoa, just go and have fun. And he did. So about 1/6 of my H index is Wei-An Zhao. Ying-Fu will tell you the same story. He then went on to do a lot of other work in Bob Pelton's network on bioactive paper because he was able to reduce to practice some of those things onto the surface of paper for bioactive paper. So those were lessons that I was learning from the people who helped mentor me about writing papers, about getting stuff out in the literature. So I wanted to just make a couple of comments. Find those mentors, right? Build those networks. For me, they were the 4 people I've already named. Science is a team sport. If you're sitting off in the corner doing something, it'll be fun, it'll be great, but the likelihood of having high impact is low because increasingly we need to bring expertise from many domains to settle difficult problems. Identify your core expertise and then broaden your horizons. So this was me in 1985. I thought I was an organic synthetic chemist, but by the time we get to 95, I'm doing a lot more silicon chemistry and some silica and interfaces. And by 2008, also biomaterials. Notice the silicon chemistry part keeps growing. And whenever you're asked a question at a conference about proteins, you go, I'm a silicon chemistry. You could pull back into the expertise set that you have and still vicariously live with experts by poking your nose into their research area and working with them. One piece of advice I wish I had taken but didn't was don't procrastinate too much. This is a picture of Alex Adranov's front cover of his 4G6 project from 1995-96, and I'm pleased to tell you the first draft of the paper is almost ready. Okay, I want to switch into something just away from research for a moment. I want to talk about teaching. So one of the things that accompanies organic chemists in particular inside chemistry and chemical biology is actually the pleasure. I really like teaching. Could you tell? I really like teaching. I try to engage audience. I like engaging students. I've probably taught 10 to 11,000 over my career because it's first year and second year Orgo. I've taught 20 different courses. I created 2. They're still being taught from year 1 up to grad courses. I really, really like it, or did, until this. This is COVID. And I want to tell you about an example that shows my ability to make really bad judgment decisions. It's March 13th. It's the last lecture we're going to give, as it turns out, not for 3 weeks, but for 3 years. Somebody is calling, it's likely spam. Okay. So some kid in the class says, we'll never see each other again. Let's all get together and take a photo. And there's the photo, which thank God was not a super spreader event, right? Can you imagine the story I'd be telling otherwise? What I did not like about teaching, like most people in the room, was this pivot. I understand why we had to do it, to online teaching. This is what you see if you go to the Teams website, all the happy people engaged sitting on the other side of the camera. That's what I saw. And if you go back 2 hours later, some of them are still there because they were never there in the first place, right? There were other challenges that came with that. We're required in Ontario to do closed captioning for our lectures. And the early days were a bit rough. So when I had in my sustainability class, Dr. Garino Sakrapante give a lecture, teams introduced him as Dr. Greeno Sexy Panty. Well, the students were pretty amused by that. Dr. Sakrapante was not quite as amused as it turns out. So this is not really suggestions. This is completely my opinion. I mean, I've done 39 years of teaching. I sort of stopped in July. That was part of the retirement deal. I think our job as teachers, as instructors, is to, in some area, in this case, mine is chemistry, teach a body of knowledge, teach some of the rules, and show how that at some level interacts with other kinds of technologies. I also think, however, as a university, that's not enough. You know, if you want to read about chemistry, buy a textbook, right? You can read about it, but that doesn't mean you understand it. I think we're supposed to be developing problem solvers and critical thinkers and people who are curious to try to encourage them. You see how important I think it is? I guess I didn't chest. Problem solvers are twice. You have to be twice a problem solver. And maybe that'll turn out functional citizens. How's that going? I'm not so sure it's going so well. So the New York Times published this piece on January 2nd this year. Half of Gen Z says they are not ready for the world. Well, I have to say, every 4th year class, when you're kind of thrown out into the world, we all feel intimidation. But employers say, well, if you weren't sure, I'm telling you, they really are not ready. They don't have the right initiative. They have no communication skills. They're bad problem solving, and they're not resilient. And I think, it's my opinion, I get to say this, you can accept it or not, we're part of the problem. So one of the things that this historical context brings is 3-hour exams have gone down to 1.5, sometimes 0 exams at all. Okay, don't you think you have to at some point be awkward and uncomfortable? It's supposed to be hard, in my opinion. It's not hard. And so things like accommodation, over 20% of students at McMaster have an accommodation, longer time, shorter time. If that was scaled, like in first year it's this and fourth year it's that, to sort of help people build those resilient skills, I'd be much less concerned about that. But the idea that, well, we have an accommodation and that's how it's going to be in the real world, that's not appropriate because that isn't the case. So I think we are encouraging them to believe things that don't actually exist. You have to be resilient. What did I do when we lost all our money from Connaught? Well, you could give up, right? Resilience is, oh, you know, expletive deleted. I guess we'll have to do something else. Students are afraid to fail. We have to fix that. The last thing I would just mention about that is the class averages are no longer on our transcripts. Did you know that? So when Johnny shows mom and dad the transcript and he got an A-, that's the worst class mark in the class. Right? We don't know. Isn't this supposed to be about, you know, transparency? How good is Johnny? Maybe not that great. Okay, I have to say one of the things I really, really like about McMaster's chemistry and chemical biology department is this fervent belief that you can do demonstrations and deliver a scientific point more effectively than just talking about it. The visual thing, the oral thing makes a difference. Sure, we blow up some hydrogen balloons. Okay, I get that. But we've done lots of demonstrations over the years. I want to try and do one in a second. I have to first tell you about the most impactful interaction I ever had with undergraduates. It took place at BSB 147. It was an 8:30 class. Of course, it was 1A, A3. And I'm asking the class some question. That kid in the front row bursts out laughing. He just bursts out laughing. I said, what's so funny? He said, your fly is down. So I pulled it up. The class lost it. So I pulled my fly up and down like this. And that was the bit. We had to stop the class because they just couldn't stop laughing. Well, that was a good thing, I guess. I'm not sure I really want to be remembered for that. Students have changed over the years. They have different levels of capacity for what they're prepared to hear, what they want to hear. I think this idea that I don't want to hear anything that I don't like is, you can have that, but guess what? Outside, that's not how it's going to be. But okay, that's fine. They've changed. I don't do the condom demo anymore. But I do think this is the demonstration that I like the best. So I have to ask this question. How many people went to summer camp? Anybody go to summer camp? How many people sang that song? Campfires burning, campfires burning. How many people? Not enough to make this work. Okay. Because you see, you're all wimps, but that's okay. No, the thing was in the 90s or the 2000s, enough kids had been going to camp that they knew what we're doing. So we actually did what I'm going to do right now. What we did, and you'll be able to see in a second, the video in the background. I took Christmas trees, just a pine tree, and it's been soaking in water for 3-4 weeks now, something like that. And then what we would do is, and I'm going to do it with a very small piece because I don't really want to set off the fire alarm and sprinklers, but what we would do, you see, is do this and then sing fires burning, drawing in the globe, and come sing and be merry. And then we do it in a 4-part round, right? It was great. Okay, what's the point? Why would you want to bother doing that? Well, what happens is if you put the pH paper into that water, it's about 5. So this is the pH paper that came out of that. We burn it, and then you take the ashes and you put them into some water, and the pH is about 9. So what's the experiment? Okay, you've got 2 different pHs. So I was in Valentino's, which is in a restaurant in Westdale 5 years ago or something, and the server comes up, you Dr. Brook? Yeah. You taught me first-year chemistry. I said, what do you remember? She said, burning the Christmas tree. What do you remember about that? She said, if you go to a forest and you look under a pine tree, very little is growing because it's too acidic. But then there's a forest fire and the pH goes up. And now all of a sudden, the pH allows new flora to populate the forest floor and regenerate the forest. Oh, That's a good outcome. I was pretty happy about that. So I think we can deliver those kinds of things in a fun way. And I've been surprised how many people have brought that up. Of course, they were bringing up the singing. That's what they really remembered, not the, yeah. And we used to do this in BSB 147, so we could really have a conflagration. It was awesome. I also want to make one more comment. This is the bad CV. This is the most important comment I ever got as, in the evaluations, our yearly evaluations. You're a good showman, you're just not a good teacher. But what did that mean? You tell the jokes, but you got to do the science. It's not enough to tell. The jokes lead to the science, right? So I guess for that person, I'd stepped over the line. Okay, I hear you. All right. Piers-Rubinsztajn reaction. So Warren Pierce from Calgary described this reaction where if you take a silane, remember that's a hydride, and react it with an alkoxysilane or a silyl ether in the presence of this boron catalyst, you make that stuff. You make a new silicon-oxygen bond. And he wrote this as a kind of warning because, you know what, if you do this reaction that you're trying to do and you put in too much stuff, it's going to go badly. And Piers-Rubinsztajn from Corporate Research and Development said, well, wait a minute, that's a brand new way to make silicones. We've been working in the area, and I wrote to Rubinstein's boss, Chojnowski, in Łódź in Poland, and said, I want to call this the Piers-Chojnowski reaction. He said, it should be the Rubinsztajn reaction. I wrote to Warren Pierce. He said, oh, you organic chemists, you always want to name everything. I don't care. He's pretty happy because there are now 115 papers with his name on it, Piers-Rubinsztajn. We named it that, and about 35 of them are ours. So we've had, this is for us a body of work. It all started with, where's Dave? There, right there. So Dave Thompson started exploring some of the reactions and came up with this really, for me, great outcome. You start off with tetraethoxysilane. You throw in that branched silicone. So you see the silicone, that blue thing has stuff on either side of it. And you can displace 3 of the 4 ethoxy groups, but not the 4th. Why not? It's a steric bulk problem, so a different utilization than in colloid chemistry. It's like the big person trying to get into the middle seat on the airplane, right? They just don't fit. But if you put a kid, so there's this little tiny SiH, a tiny thing, it squeezes in, no problem. So in 2 steps, we were able to take that molecule and make this molecule in over 90% yield as a single compound. That was unheard of in the day as a silicone in silicon chemistry. Highly branched, highly structured things, especially when it was relatively simple. So we tried to milk this process. Shui Liang showed that you could have, for example, 2-fold symmetry. We'd put a brush on the bottom, a brush on the top. Then we could change the chain length in a very easy fashion. We made libraries of these things. Some of them are useful tackifiers in silicone rubber. They make them sticky on purpose. But the best one, I think, in an aesthetic sense, is this one. And that was done by John Grande, who's sitting right there. So John took this same old tetrathoxysilane. So 4 alkoxy groups became 12 alkoxy groups, became 12 vinyl groups, became this giant ball, this dendrimer, which is now almost 14,000 grams per mole. That's a big molecule, pure. Now, people had made dendrimers like that before. 0.5 nanogram, 3 nanograms. John could make, if necessary, a gram. It would have taken a time, but we could do it. It's about showing that the power of this reaction for highly branched materials is important. But there are other ways to utilize this reaction which are completely different. So Vinod, I think, did this. Where's Vinod? Vinod's here somewhere there. So Vinod did this early work, I think, where we took the silicone, this different blue and red silicones, and put them into the cheapest printer we could buy at the time, which was 25 bucks on purpose. The cartridge was more expensive to replace than the actual whole printer. And we put one of the ingredients in one chamber, the other ingredient in the other chamber, catalyst in the third chamber. And what we did was to use this printer to print it. It's a bit hard to see. There's the starting paper. You might see some fuzzy stuff, which I'm now going to color in red. That's silicone. If you put a drop of stuff in this side, it will try to migrate to the other side. And that became the basis of a bioassay on paper, but it hinges on the fact that those red silicones act as a barrier so that the stuff doesn't go everywhere. It's channeled to go where you want it. And Medea Khan, who's sitting there, took that to Kenya to show that you could do bioactive paper in a resource-deprived economy and do real chemistry. That's why we chose the cheapest one. The other one was Jason Zhang, who said we can do the same kind of thing with lignin. Lignin is the brown stuff in trees. And so if you do a Piers-Rubinsztajn reaction, some of the phenols that are there will react with the silane, and you make a piece of silicone rubber. It smells exactly like, you know, a fire. It smells like somebody's had a fire. And lignin is normally used to run the pulp and paper plant. It's often burned. It's considered waste in many situations. And Jason came to me and said, I know, we'll put 35% wood in the silicone, and it will be better as a flame retardant material. Now, I don't know about you. My skepticism was pretty high. I said, sure, go show me. So he did. This is a pure silicone. It burns by itself for 120 seconds. If you expose it to 10 seconds of flame and pull it away, the stuff that has 35% lignin in it only burns for 25 seconds. Why? We think because as you're heating the whole system with the flame, some of that heat is absorbed by the wood as it's charring inside, and that allows you to build a better silica layer, which is an insulator on the outside. Doesn't matter. Let students try stuff. They might be much smarter than you. Okay. And I've been a benefit of that over and over. Well, here's a second vignette. I want to talk about the administration. It's my last shot, right? My sense is that some of you will know the story about Don Quixote from Cervantes, this guy who would go out and would essentially sort of tilt at windmills because he thought they were giants that need to be slayed. He saw things a different way. Sometimes I think he saw them the right way, but most of the time everybody said, you're doing it, you're nuts. There will be administrators who say, I should really be Mike Quixote because for my whole career, I have made suggestion after suggestion after suggestion about how I think we could do it better. For me, but for all of us, right? There are just ways to do that. So I'm going to give you some examples of things I think that the administration could do better, which would be great for the university. The first of these is to just be more transparent. I'll talk about this more in a second, but if we don't know what you're doing, we can't get behind you. Does anybody know where we're going? I have no idea where McMaster's going. What's important? I really don't know. Maybe it's because I'm out of it or I'm not interested. We should all know. And we can either get on board or not, but we should know. 2, don't lose connection with the people that you work with. I'm going to talk about that too. There is this huge strata of people that have to support us and make us work. And I actually think that they should be doing, in the first year of a senior administrative position, undercover boss and go work with the cleaner and the accountant and find out what they are really suffering from. Implementing policy. The number of times people have told me it's against policy, that is just a red flag for me. I have a feeling this is how it happens. Look, these people who are administrators, they're nice people. They're doing their best. They're seeing the world in a different way. And I kind of have this sense that I'm saying to somebody, this is bullshit. And then it goes to my chair and to my dean and to someone, and it arrives at the provost going, oh, this is the fertilizer that is going to support what we're trying to do. No, it's bullshit. But you don't know that unless you talk to me, right? So, hypocrisy is a moral buzzkill. I have had a lot of senior administrators complain. Not report. Reporting is fine. Complain. Research costs us money. Stop doing it. Don't expect to be the 4th most research active university in Canada, but just stop doing it or stop complaining about it. And those are little things, but they drive me nuts. Maybe listen more to your hearts and minds and less your lawyers. Every decision at every meeting of administrators all the way down from the top to the bottom should answer 2 questions. Is this making teaching better or is this making research better? Because so often we're doing stuff like report on activities on electronic forms, which is not helping anybody actually. Finally, do not touch, and I think this is the one thing that I want to say, and I'll say it twice. The most important thing about McMaster is the collaborative spirit. Dave Emsley sold me this catalyst that I couldn't get otherwise because he was just prepared to do it. He's prepared to help out. 3 times in 39 years, people have said, I'm too busy to talk to your student. 3. You'll see how many people I've co-published with because we just, let's try it. Don't touch that. I want to talk about our mission statement, but I need to give you to preface it by where my students come from. So this is where the postdocs came from, PhD students, master students, and of course, my undergraduates are not all up north of the Arctic Circle. I just couldn't figure out how to put them in. So they're from all over the place. So that's one of the things. So that's my entire group over the entire year. These are people who spent at least 3 months with me. And that is the male-female split. And so that's cumulative on the right-hand side. So it's 57% male, 57-43, which in a chemistry department, I think is pretty good going. I am so lucky that in the early days, Canadians wouldn't work with me. Oh, you want to work with me? Let's go. And what you learn is all that stuff about diversity and different perspectives and cultural backgrounds and training. It really works. You throw people in a room and say, let's go, and they will. And they all bring what they want to it. So let's look at our mission statement. Inclusive excellence. That's a nothing burger. What the hell is that? It's a nothing. It doesn't mean anything because EDI is a verb. It's not a noun. It's a thing you do. It's an action statement. I think they should be inclusive teaching and learning, inclusive research. Bring everybody into the pot. That's my opinion. That's how we have to do it. It's not that EDI is not important. It's incredibly important. And I have seen it long before EDI was a thing, right? I was really lucky. And often people don't get that chance, but I just happened to, as you saw from where people come from. 4 people I want to talk about as administrators. Mamdouh Shukri was the Dean of Engineering. He helped me out of a very sticky situation as a scientist. He had no obligation. He was just a really good guy. Harvey Weingarten was the most transparent, by far, administrator we've ever had. He's the guy who said, this is where the train is going. This is where I'm going to put resources. If you want to join me, that's great. If you don't, have fun, but don't come. And we knew the rules, right? That's all I'm saying. We knew the rules. For 15 years, every faculty member gets a letter from the president every year. Here's your salary. Peter George crossed out Dr. Brook and wrote, Dear Mike. It's so little, but it's not so little. It says we're all on the same team, guys. And that's not the only thing he did. It was about trying to build a community. We got this unbelievable community here. We've got to really try hard to take advantage of all of the things people bring to it. And the best administrator I've ever worked with is sitting right there, Jill Goward. Just bar none. Okay, I talked about administrators, talked about teaching students. You've seen some of the work of my research students. There's more about that later. But we kind of forget this whole place runs because there are people out there doing stuff. So the Dean, Maureen McDonald, I had a conversation about this. You have all these people who are overworked, underpaid, kind of not treated often very nicely, and they just beaver away to make the joint work. We are so lucky, huh? And I only picked 5. So Leah for her work on safety, Josie Petrie in the chemistry, at that time it was just the chemistry office, Gloria McCleary in accounting in Rhodes, what is now Rhodes. And the last 2 I left, these are kind of the most important because they deliver 2 things. First, real excellence in making the place work and in changing the environment. So Marg for, I don't know how many years was it, Marg, like 25 years, just out, where's Marg? There, she's right there. Just down the hall, 10 offices away. I could hear her laugh at something. Do you know what that does to your day? It makes it great. And Gloria, the cleaner on the top of ABB for 3 years, I think, the same thing. She made me dress up as Fisherman's Friend for Halloween one day. It changes the dynamic. And I am incredibly grateful because all of those people help us get through things. Okay, I want to finish off on the last research vignette, and that's about sustainability. Paul Harrison invited a guy named John Warner who wrote a book called Green Chemistry in 2017 or something, and I had an epiphany. In other words, green chemistry is a list of principles about how you do stuff that actually causes less damage to the planet. It is the equivalent of the Hippocratic Oath for doctors. First, do no harm. Like, it's not that dumb, right? Like, it's obvious we should be doing that. You just have to look outside to know there are things we could easily do. And chemists have some of the responsibility, of course, for creating that harm. And we've got the tools to remediate. So what do we need to do? Use stuff over and over and over before it ends up in the environment, right? That's an obvious way. And so the first thing I want to talk about is exactly that. So everybody right now has, as their most abundant protein in blood, human serum albumin. It's got 7 cysteines. The cysteines have that little green thiol there, but 6 of them are bound up as disulfides to give the protein its tertiary structure. The body happily pops back and forth between the 2. It's a very easy process to do. And we asked the question, could we do this with silicone elastomers? So there's a silicone with a thiol on the side. You oxidize it to get the disulfide, and you get beautiful elastomers. They're perfect. They function just like you would expect and just like you'd like. But the Piers-Rubinsztajn reaction reduces them, and that's what you're seeing in this video, back down to fluids. And there are 3 reactions that take you from an oil and back to the exact same oil, and the yield is 95% 5 times in a row. I think it's probably the most recyclable silicone on the planet. So we can do things like that. You can also apply chemistry in one area to another. So tires, rubber tires, I think could be argued to be the most egregious use of single-use plastic or rubber or something on the planet. They're held together by sulfur-sulfur bonds. So I'm the carpenter with a hammer in my hand looking for a nail, right? Oh, sulfur bonds, let's go kill some. All right, well, worked great. So there's Scarlett Zhang taking the tire, grinding it up. We put it into a little reaction flask for an hour, and then you could pull out by a magnet the metal, all the other stuff, the catalysts and fillers by filter paper, and you get over 90% back of the oil that can be used to go back into a new rubber. That's sustainability. We're closing the loop. We're building circularity into the product. It's too expensive to practice, but okay, at least it shows it could be done, and that's the kind of thing that's very attractive. Natural products, if you take a silicone, high carbon footprint, but if you can get the same level of performance and dilute it out with a natural product, that it would be a good thing. And we decided to look at hydrogen bonding. You know, chemists don't often think about hydrogen bonding, but it's holding all our DNA together, so it must be okay if you have enough of them. So we took a silicone with an amine on the end and reacted it with this glucose. It's called gluconolactone, so a little bit of sugar on each end. Before you put the sugar on the end, it's a viscous oil. But once you've put the sugar on the end, it will absorb the energy of a small ball or this big ball, which was a bit of a surprise to us because there is 0.1% sugar in there. Nothing. But if you wait 15 minutes, they sink to the bottom. They're not a rubber. They're a temporary rubber. So then we did this a different way. We didn't use a long chain. We used a shorter chain, same amount of sugar net, but we dissolved it in a low viscosity silicone fluid. And at 10% loading, it more or less absorbed the energy. But at 30% loading, it basically gave back all the energy to the ball that was dropped from about a meter. But once again, if you wait half an hour, it's a fluid. These are working fluids, like clutch fluids and things like that, based only on a teeny bit of sugar in what is otherwise grease. So that was a nice outcome for us, that natural products can deliver special things. Robert Bowie, who's there, showed that we can expand this. So he took vanilla extract. His reactions always smelled so great. Like you walk in, it's like somebody's baking cookies or something like that. They look horrible, okay? Like this is not the kind of silicone. People want clear silicone elastomers. They don't want these black ones. But if you take vanilla and dimerize it, so you have these aldehydes, that carbon-oxygen double bond reacts with the nitrogen to make this imine bond to make rubbers. And what was interesting, aside from smelling great, is that when you did it, you made these rubbers derived from vanillin, you ended up with much better performance than if you used the complementary materials derived from petroleum, because it was delivering extra benefits for reasons I don't have time to get into now. So yes, we diluted the amount of silicone, and we also had good performance, and most important of all, they're meltable. They're thermoplastic elastomers. So you heat them up to 150 degrees, melt them, and reuse them in a new form. So it's kind of circular in that sense, or reusable. Sorry, it's reusable. And the last one I want to talk about is gelatin. How many people have made Jell-O with their mom at one point? Yeah, okay. So you know how to do this. You just basically cook it up with water. What you don't normally do is throw in an amino silicone and formaldehyde, right? So formaldehyde's an embalming fluid. It's not the greatest stuff. It's toxic. But if you do that, what happens is, like, silicones don't dissolve in water, but they will under these circumstances. And that little red link there is showing that the formaldehyde is bridging a silicone to gelatin. Well, why do we want to do that? Because we could then dilute out the silicone number one. And they're not the most beautiful things. Some of them, more gelatin, the whiter they are, the more silicone, the clearer they are. We can control the cross-link density. But the most important thing is that once you've got the gelatin, you get it to fall apart. How many people, when they made gelatin, tried to add fruit to the gelatin? Anybody? What fruit do you never add? Right. Why? Because pineapple degrades gelatin. And so what happens if you take those elastomers and put it into a mixture of actually bromelain, which comes out of pineapple stems, in about a week, you have gelatin dissolved in the bottom in the water and silicone oil floating on the top. And silicone oil degrades on its own in the environment. It hydrolyzes in the summer, not now, in about 3 weeks, and it goes back to sand, water, and CO2 in about 2 years. So we have a biodegradable material, which, by the way, is not only working in the lab with the Hamilton composting facility down near Stelco. What we were able to do was take materials that were based on cross-linked silicone, put them into their industrial composter, and 1 month later, nothing is left. It's all gone. So these are the first compostable silicones. Hopefully that's the next paper on the list that we were able to make. So we started off, I wanted to show you this picture of my expertise. Yeah, it doesn't look like that. You saw those ones. This is me today, okay? So I really know a lot about silicone. I really know a lot about silicon. I know a lot of bits and pieces about other things. I followed my nose. I followed my curiosity. I'd encourage you to do exactly the same thing, because often you have the solution to the problem that that person is struggling over, because you just have a different expertise set, and it's fun to work together. Okay, let's do the numbers. We're all scientists. What's the data say? The data is 317 and 1 submitted paper and 15 chapters, a whack of presentations. I wrote a book, which in the day was important. I mean, people don't think about that so much. It was a snapshot of what the silicon world looked like in the late 90s. I would really like to thank Wiley for publishing it on December 28th of 1999, rather than waiting for the new millennium. Thank you very much for that, but okay, that's what they did. I've spent about $12.2 million to do that. So if you do the math to take 12.2 divided by 317, that's what it costs per paper. Was that good value for money? You tell me. I got money from a lot of people because I couldn't get enough from one person. So you just beg until they give you enough money. And I want to thank these people. And there are actually several that refuse to allow me to show their logo because they want to keep it quiet. Well, that's fine. I am incredibly grateful to them because they enable hiring undergrads, grads, postdocs, running the lab, all that stuff. And top of the list has to be NSERC, who, by the time my current Alliance grant runs out, will have given me money for 48 years in a row. Okay, recognition. Okay, McMaster. I was really pleased to be Faculty of Science teacher for a couple of times. International and national awards. Of course, these are great. And they represent the contributions of the team. That's wonderful. But actually, where I'm sitting right now, that's not that big a deal. This is what's a big deal, is this ability to work with all the people that I've had the opportunity to work with. Like, those are the people I published with just at McMaster. You see how little I like writing papers? Get somebody else to write them. It's much easier. No, but true. In the chemistry department and all in pathology, in Chem-Eng, in Ravi, as in mechanical engineering. And these two guys, we just got an Alliance grant. They're in civil engineering. So next year, I hope their name will be on this list. These are people that I've published with in the rest of the world. It opens doors. It's fantastic, this ability to collaborate with other people. I have to make a comment because that's the Swiss Army knife that Dan Chen, where's Dan? Dan gave me in 2000 when he arrived as a postdoc. He too is, we're all getting older together. He's been working as a research scientist for over 20 years now. He is a Swiss Army knife, okay? He can do anything. We celebrated his contributions when he had a 20-year anniversary, which is tough because we're not allowed to hire technicians in chemistry on your NSERC grant. You got to find other money for that. And it's been such a great collaboration, such a great ability to work together. And now we're involved in 2 startup companies. These are the undergraduates in 4 time periods that I've had the pleasure of working with in my lab for at least 3 months. I'm sorry that your name, Daniel's going, wait, I wasn't up there very long. Okay, no, you're not, sorry. On the left-hand side are master's and PhD students. On the right are postdocs. Bold names are professors, if that matters. 280 individual different people who spent at least 3 months in my lab. It's a miracle. It's just fantastic. How lucky am I? And what's interesting is that it goes beyond that. If you look at those 4 ladies right there in Berlin in 2014, now you can see them picking apples with their kids last September. You form these long relationships. It's a community of scholars, but it's a community beyond that. These are 4 of my Chinese colleagues from the late to the teens that met together in Hangzhou. This is Dan and me and Paul Zalisco, who's there, and 2 other students from around the 2005 era. Yeah, this is Dan and me looking pretty stupid, but anyway, and that was my birthday. But it's these long time collaborations that allow you to do things you couldn't otherwise imagine. And then I have to thank my wife and my kids because occasionally I complained, Like referee to that student, this administrator, that colleague, like every night at dinner for 39 years. So the support I've received from all of them is just really, really special. Okay, everybody who's been at this university has almost certainly done a titration, right? And so that means you know how it works. You take an unknown, you throw it into a flask, you then add slowly, hopefully you don't add too much, but you slowly add all the stuff that's needed from a known amount of material, and you do this titration, right? Well, the titration allows you to get from the unknown to the known, and hopefully you do it at the right time. In a perfect world, you get to the equivalence point, but in a practical way, you get to the end point. We measure something, we see a change, that's the end point. And there will be people in this room who say, yeah, you're leaving too early. And there will be people in this room saying, are you still here? Are you leaving too late? But what I want to say is that there are three classes of people, kinds of people in society are allowed to wear gowns: academics, and the judiciary, and the clergy. But for me, this is the end point, right? So this is the time to stop professing and take this off because it is really hot. And then to say, if I stand here and I look back at what we've been able to achieve, it's through all of the efforts of the people listening and the people in the audience and all the people who are cleaning the floors and everything. It's the team spirit, and that has made my career kind of magic. So I want to thank all of those people for all of that support. I would like to thank you who came on Zoom and you are here for coming to the show. And I want to thank the university for taking that bet 40 years ago. Thanks very much. Wow. Oh, wow. Okay, wow. Okay. Now, it's customary to allow questions to be posed, and I would be certainly willing to give you more suggestions for the administrators if you like. Or I don't know if there are any questions. You're more, yes, Bob. It's a big number. It's a really big number. The first rejection took place in 1989. I can tell you exactly what it's about. The last rejection took place in June. I have written really a lot of letters. I can't count them. Carlos. It's a very fair question because so sometimes you do things, and I certainly do things where I boil over and overreact. I have learned not to send the letter at that moment. There have been a couple of letters that I really wish I could pull back. Maybe, I don't know, 5 or 10. I really wish I have a second shot at. It's unfortunately more than 0. Yes. Oh, thank you. Wow. So in silicon chemistry, I think, I talked about this giant footprint, carbon footprint. I just wrote a review, we'll see whether it's accepted, in which I basically lambaste the silicon companies for not funding work in the direct process. Because if they would spread a little money around and take very different perspectives, we might have a chance to solve what is an incredibly expensive problem. So that should happen. I don't believe it's going to happen. And I think they will be dragged kicking and screaming into the kind of sustainability stuff we talked about because they're going to be forced to. I know that they are already worried. They're already buying carbon credits. So they all of a sudden like recycling because if we do recycling, that's a carbon credit. So I guess I don't know the answer. Of course, my bias is that this whole green sustainable thing, which is being widely adopted by more organic type polymers, is going to be adopted by the silicone polymer chemists. But there aren't many academics doing silicon chemistry. So I don't know. It's a good question. Oh, that's a great question. So someone said, someone wrote back to me, I asked me for a master's position yesterday. I wrote back and said, well, I'm not taking any more graduate students. Enjoy your relaxing time, so she wrote back. Well, I hired 5 new undergraduate students in January to accompany the 2 I already had, one and a visiting student and grad student from Denmark and Dan. So there are now 8 people in the lab, which is more than we've had in the last year because we got money. It's as simple as that. No money, no people, right? We're going to focus in the next while on the startup company that we're doing, which is taking your old Michelin tire and putting it into silicone rubber. And the idea is it's a waste. The tire is basically waste. We can maintain performance of the silicone, but we can drop significantly the carbon footprint and we can drop the price. So in North America, we can drop the price. Nobody cares about green. In Europe, they really want green, so we're going to focus on that. So for us, it's about trying to do what I talked about in sustainability, but in a very narrow, focused, practical way. And we're doing some crazy stuff too, so it's fun. So the answer is I'm not really retired. You know, he hasn't left the building yet, so. Okay, I got to stop there. I want to thank again everybody for coming. Really, it's so great to see so many faces from so long ago. Is Mo here? Mo Hattie? He was going to try and come from Windsor. So he is the 3rd student I had. The first 2 are dead. So that was kind of amazing that he wrote. And I've had people, the response has been amazing. It just shows that this community, yeah, it can happen, right? Like this, and bring it on, because that's I think what you're going to remember. Anyway, thank you very much. Appreciate it.